Although a relatively simple molecule, DNA is responsible for every protein produced throughout your body. It is therefore important for the replication of DNA and production of RNA to be as efficient as possible. But first, what is DNA? Deoxyribonucleic acid, or DNA, is made up of smaller monomers of good nucleic acids, which themselves are made up of three constituent parts. The deoxyribonose sugar and phosphate, which forms a backbone that provides the framework for the third component, nitrous bases. There are four bases: adenine, thymine, cytosine, and guanine. Two nucleic acids combine by expanding water through a condensation reaction, forming a phosphodiester bond. Additionally, each base has a complementary base in which it can readily form hydrogen bonds. Adenine with thymine and cytosine with guanine, which binds two strands together. During cell division, a new set of genetic material is required. To accomplish this, DNA undergoes a process of semi-conservative replication. First, the enzyme helicase breaks the hydrogen bonds between the bases, unzipping the DNA. Next, three nucleotides are activated and bind to their complementary base. The nucleotides are then bound by the enzyme DNA polymerase, forming phosphodiester bonds. This forms two DNA polymers. Both are a strand of the original DNA molecule and a new strand made from three nucleotides. DNA encodes for proteins, but it must be transcribed by the polymer mRNA. mRNA differs from DNA in that it encodes only for one protein, is single-stranded, and has the base uracil instead of thymine. mRNA is produced through the process of transcription. An enzyme acts on the area that encodes for a protein, exposing the DNA nucleotides. One of the DNA strands, the template strand, pairs with three nucleotides present in the nucleus. The enzyme DNA polymerase then polymerizes the nucleotides, forming pre-mRNA. Only 12 base pairs can be exposed at one time to prevent risk of mutation. When RNA polymerase reaches an end codon, it detaches, and the mRNA molecule exits the nucleus via a nuclear pore. tRNA, or transfer RNA, is responsible for synthesizing polypeptides using the code from mRNA. First, a ribosome becomes attached to the starting codon AUG. The tRNA molecule, with the complementary anticodon sequence, pairs with the mRNA codon, carrying a specific amino acid. This happens along the mRNA molecule. The amino acid on the tRNA are then joined together by a peptide bond, by using ATP. The first tRNA molecule is released from the amino acid. This repeats up the chain until a stop codon is reached and the polypeptide is released.